Because it's very difficult and expensive to collect these samples from patient, there are models that people can use to simulate a human tumor. These include cancer cell lines. Initially, this, for example, HeLa is a very well-known one from cervical cancer. So you take the cancer cell from the patient tumor and you plate them onto plate. In the early days, people didn't have good success. HeLa was one exception. It was just so growing so fast that it was able to grow in the petri dish. But over the years, people have learned better ways to treat the cell, grow it in different media, so that there are a couple thousand different cancer cell lines available now. Another is called tumor organoid. If you take the tumor from the patient and you grow them in kind of a three-dimensional, they grow into tiny little tumors, whereas in cancer cell line are usually grown as a two-dimension in the petri dish. And so in this little organoid, usually it can mimic the drug resistance better than the cancer cell line in the two-dimensional scale. And sometimes in recent years, people are also able to grow the organoid still with the immune cells or fibroblasts near it, whereas cancer cell line, you are only dealing with the cell lines and not the overall environment of the tumor. So these are models. Another one is a patient-derived xenograft. So this is initially from the patient tumor, and you take out the tumor and you chop them up into smaller pieces, and then you implant them in mice. These are not wild-type mice. They are often nude mice, which means that they're immune compromised, and that's how the tumor can grow in the mice. And sometimes there are studies like this where after the patient has surgery, the tumors are taken out and they can be given implanted in multiple tumors, and scientists or doctors can treat the mice with different drugs and then see which tumor can shrink in the mice. Then they will know which drug to give to the patient. In terms of growing, the patient-derived xenograft is the easiest. Most patient tumors can be grown in mice. Cancer cell lines is a little harder organized, I think probably even harder. And also there are genetically engineered mouse tumors. So for example, scientists can create mutations either using a carcinogen or specifically create a mutation on a gene, and then this tumor will evolve. And once this tumor is there, you can take out the tumor and then plant them in new mice, and they will also grow the tumors carrying this mutation. And so these type of models are much easier for scientists to test many different genes, many different drugs, do manipulations and figure out the response mechanism. And so one example, by the way, these type of mouse experiments are still very expensive. And so most of the data available are still on the cell lines. And so in order to do this cell line analysis, usually we can grow these cells in petri dish of either 96 well plates or 384 well plates. And then each well, you can add a different drug. And these are usually done with a robotic automation. And then we can measure the phenotype, such as growth. You can also measure the gene expression of the cell after the drug treatment, which will then give us a lot of very interesting insights. And depending on this, the scale of the screen, there are two types. One is you can test, thousands or even tens of thousands of drugs on a few cell lines. You just want to see, this is to see, what the different drugs are doing. For example, the Broad Institute have a project called Connectivity Map. They measured expression of only 1,000 genes. They have probes to just measure 1,000 genes. And after the cancer cell lines, they have many cells, like a handful of cells that are treated with tens of thousands of drugs. This can help them, and then after the drug treatment, they can see the cell growth, but they can also measure the gene expression and by the value of those 1,000 genes. And based on the 1,000 probes, they can look at all the public data and infer the remaining 20,000 or 19,000 genes, or 20,000 genes in the genome to get a rough idea of the other genes, how they change. And so using this, for example, you can calculate the differential expressions of these small molecules. They can group the drugs into different classes because there are small molecule compound libraries that can have, sometimes millions of different compounds. You first want to see roughly what they do. And so using this type of cell line, like a small cell lines, number of cell lines, if you can measure like 1,000 genes, you can get a rough idea. Okay, this is one group of drug, the cells respond in this way. There's another group of drug, the cell responds in another way. That's kind of different from the first class. And interestingly, there is one study. This is a study from the same group that developed the connectivity map, or the L1000 gene panel. They look at the TCGA between tumor and normal for some cancer type. This is neuroblastoma. They look at what genes are differentially expressed, especially upregulated in the cancer. And they ask what specific small molecule can treat, when you treat the cancer cell with this drug, the downregulated genes are the upregulated gene in this cancer type. So basically you find a small molecule that can have a maximal reversal of that disease signature, meaning that if you have this type of cancer patient, if they are treated with this type of with this small molecule, those genes will be now downregulated and potentially be used as a cancer drug. And so you can see here now, using these cancer cell markers, cancer cell lines and these marker genes, you can really identify a new drug for a disease, right? Another type of screen is we use limited number of drugs, sometimes even a single drug, but you test in many cell lines. For example, if you know that your drug is inhibiting a gene such as EGFR, and you want to see what cancer cell lines can respond to this, then you can take, so there are projects like this, 3 different projects, CCLE, CTRP, and GDSC, and they have hundreds of different cell lines. And they are treated with different compounds. These are usually compounds that people are interested in developing into a drug. The target of those compounds are usually known. And after the cells are treated with the drug, we usually don't, in this case, measure gene expression change. Instead, we measure the sensitivity in terms of IC50. So by the way, the cancer cell lines, the hundreds of cancer cell lines have already been comprehensively profiled for mutations, for gene expression, and recently a subset of those cell lines have also been profiled for epigenetics and proteomics. But all of them have expression and mutations done. And so after you treat the cell, so usually this is done in not just one drug, one cell. You usually treat the cell with the same drug at multiple concentrations. And so, sorry, we don't have dots in here. So theoretically, you should have, when the drug is this concentration, this is how much this cell is growing. And when it's treated with for a concentration from low to high, this means that when the concentration of the drug increases, eventually a lot of cells will die. For example, salt is not really a cancer drug, but if you treat the well with a lot of salt, eventually those cancer cells will die. And so you will generate a curve like this. And so in this 386 well plate, some cells will, in one well, the cell will be treated with low concentration, medium, and then that next one will be medium concentration, next will be a higher concentration, and the next will be the highest concentration of the drug. And after that, we look at the overall cell growth, and then we come up with this curve. So there is a term called IC50, which is the concentration of the drug, which can have the half of the maximal inhibitory level. And so, you can see here, it's like, what is the the maximum inhibition here, what's the half of that inhibition level? This is the concentration. Another way is to look, because the number of points might be limited, sometimes there could be individual noise on one concentration. And so people also use area under the dose response curve. So basically the overall area under this curve as a measure of drug sensitivity. But in general, whether it's IC50 or area under the curve, the smaller the value means the cells are more sensitive to the drug. For example, if the curve drops very, very quickly, your area under the curve will be small, meaning that even at a very low concentration of the drug, a lot of the cancer cells are dying, right? And so using this approach, because these the cancer cell lines are well characterized already, for a particular drug, we can start to develop response biomarkers. For example, this is a drug, lapatinib. And if we look at lapatinib is a HER2 inhibitor. And so if you look at the IC50 of the hundreds of cancer cell lines, if we notice that if HER2 is also called ERBB2, the ERBB2 is the official gene name. You can see here, when the HER2 gene is wild type, the IC50 is very high, meaning the cancer cells are not very sensitive to the drug. But if you have a HER2 mutation, the IC50 become much lower, meaning the cancer cells are now very sensitive. They're dying with this lapatinib treatment, right? Another example is FGFR2. Again, the wild type, the cancer cell lines with wild type FGFR2 are resistant to the drug, or at least they're not being killed by the drug, but the cancer cell lines with this mutation are much more sensitive. Another one, this is MDM2. This is really targeting DNA stability. So there is a drug, Natlin 3A. You can see the wild type P53 cells are sensitive, but the P53 mutant cells are actually more resistant to the drug, because with P53 mutation, the cancer cell can be much more mutated, DNA instability, they can develop resistance. Yeah, and so on. So you can see here, based on the mutation profile, you can look at the response biomarkers to say, this drug should really be used to treat patients with certain mutations. In addition, we can look at gene expression. This is another example on the IC50. Well, this is the dose response curve. And they only tested on, say, in this case, limited cell lines. And what you can see is that there is a gene called AHR. And we are drawing the cell lines based on the expression level of AHR. You can see here, when the AHR gene expression is high, this cancer cell line has a faster kind of a area or smaller area under the curve. They are more sensitive to the drug. But if AHR expression is low, it's less sensitive to this MEK inhibitor. We can also use this type of approach to identify novel vulnerabilities. This is an example that was published in Nature by Ron DiPino's lab, because sometimes the cancer cell growth needs parallel pathways in here. So for example, in order for the normal tissue to metabolize glucose, it needs enzymes, either ENO1 or ENO2. Either one of them are okay. Interestingly, in glioblastoma, this is a brain cancer, no cure. Average patient survival from diagnosis is only less than a year. And interestingly, it was discovered that 5% of those tumors contain a ENO1 deletion. It's not even a driver gene, it's a passenger gene. It's next to another tumor suppressor, which is frequently deleted. Well, not frequently, but recurrently deleted in about 5% of the glioblastoma gene. So without the ENO1, the cancer cell can still grow because it still has ENO2 to help the cancer cell metabolize glucose. But interestingly, ENO2 has a small molecule inhibitor. And when you treat the cell with this with ENO2 inhibitor, the normal cells are okay in this case because they still have ENO1 to help metabolize metabolize glucose, but the cancer cells will die because it does not, the deletion is the cell does not have ENO1 anymore. And so the ENO2 inhibitor would work really, really well. And so if, from enough cancer cell lines with glioblastoma, we can identify mechanisms like this, right? So model systems are very important. Unfortunately, there are also some pitfalls for compound screen. A lot of these initially are associations. We're seeing that this mutation is associated with a response, or this gene expression is associated with a response. There still needs to be additional tests to really validate that this is really causal and understand the mechanism. And also, the cell line lineage representation may not be sufficient. Even with 1,000 cell lines, sometimes you still don't get a good enough representation. And if you really want to see what happens in a mutation, in this case, the FGFR mutation, out of the hundreds of cell lines, there are only 2 that are mutated. I think if you only have 1, you just cannot even be confident that this is a real mechanism. And also, if this is a acquired resistance that cancer cell develop later, you may or may not see it. If the cancer cell line already have that mutation, you can find it. But these cancer cell line experiments are usually done in a matter of 2-3 days. So some of the later, like developed resistance mechanism, you may or may not be able to find it. And finally, it does not consider immune response because the cancer cell line growing in Petri dish does not have immune cells nearby. So we lose some of that information. Therefore, there are also things like genetic screens. If you can get rid of a gene in a cell and see how without this gene, how it influences whether the cancer cell can still grow, or whether, or how this cancer cell respond to a particular drug. And of course, if you just get rid of one gene at a time, it's very time consuming. Can you do this in a high throughput way? Knock out every gene in the cell in different cells and do the whole thing in one big experiment. This is called a genetic screen. And we will discuss CRISPR screen later in this module.